Thank you.
So thanks everyone for coming. So I'm very excited today that we got Dr. Greg Gloor to come. He's the chair of the biochemistry department at the University of Western Ontario. Greg's had a very, I would say, aspirational and cutting edge career. So to put this in perspective, Greg's PhD thesis was sequencing the mu bacteriophage. And when I say sequencing, I mean, I'm pretty sure running gels and counting the bands across the gel to sequence the entire phage, which, fun story, was defended the year I was born. Now, Greg went from there to do a postdoc at UW-Madison, where he was working on a lot of Drosophila research and I do believe tools for genetic engineering in Drosophila. He took that from Memorial University in Newfoundland and then eventually back to the University of Western Ontario, where he's been since. One of the big reasons I think for having Greg come is I think he's an amazing scientist and great speaker, but he also has an uncanny ability to be about 2 to 3 years ahead of what everyone else is doing in the field. And we can look at a couple exhibits of how this is true. So I think in 2011, Greg published, I think probably fairly the first paper about doing amplicon sequencing on Illumina, which I think is 2 years before the Earth microbiome protocol. Greg was doing, I think, meta-RNA-seq or meta-transcriptomics in around 2011. So, I mean, let that sink out. And when going through that data, I think he realized that, microbiome data sets are compositional. It's not optional, which is also the name of one of his manuscripts, which I think should almost be mandatory reading for anyone in the microbiome. I think he can talk about these concepts as it goes on. Among other things he's done, he was involved in one of the major MAG papers that was published in Nature a couple of years ago, which has really been a game changer in the gut microbiome field, as well as a number of wet lab experiments. He's not just a computational person. And so now I was really excited to see that Greg is going to be talking about meta-transcriptomics today, because quite literally I was talking to someone at Hershey last week and I said, really cool technique on paper, but good luck trying to find anything actionable or informative from it. So the fact that Greg now is working on methods to do meta-transcriptomics on microbial communities tells me that we should start doing meta-transcriptomics ourselves so that we're up to pace with the leaders in the field a few years from now. So with that, thanks so much, Greg. And I'm wearing back to the microbiome center.
I'm already wearing the buttons. Well, thanks for the invitation, Erica, and thanks for the introduction, Jordan. And really what it is I have a super short attention span, so I just get bored super easy. So I titled my talk Meta-transcriptomics ready for prime time because I'm still not sure. And yes, we published a meta-transcriptome paper a number of years ago, maybe a decade ago now, and we've been sitting on the follow-up data set for almost as long, and I'm going to present a little bit about that one, mainly because every time I go back to it, I think, this is still not right. I still don't quite understand it. So, and I apologize, this is hockey, not football. I just ran out of time to find a nice, a good football analogy. But, the goal is if you have a meta-transcriptome data set where you're trying to collect gene expression information across all the species in an environment, we want to do a few things. So the easy one is identifying taxa that are driving the difference between groups. We can do that with meta-transcriptome. We can do that with 16S. We can do that with metagenomics. That's fine. The next one is identifying genes and functions that contribute to that difference regardless of the taxa. And this is where it gets really tricky, okay? And where it gets really stupidly tricky is when you're doing any analyses. Any analysis that you do, you want to make sure that it's internally consistent and not driven by the bioinformatic method, but by the biology, okay? And a lot of stuff that's published is driven by the bioinformatic method and not by the biology. So I have, why is it hard? Okay, what makes it hard? Well, so first off, the species themselves can stay the same at abundance. They can change in abundance and they can change in occurrence. So in this example, I have 3 bacteria, bluish, reddish, and purplish. I'm a man, red, blue, green. That's all I see. I'm sorry. And so they're changing in occurrence. Group 1 is missing the purple one. Group 2 has all 3. The genes, the changing in abundance in group 1, the red one is less abundant than the 2. And then there are also the genes inside are also changing. So there's a huge number of moving parts to start with. Okay. The second reason is that we actually don't measure the moving parts directly. Okay. Using high throughput sequencing is kind of like using golf clubs to play golf. You know, they're poorly designed implements for the task at hand. Okay, there's like one golfer in the room. So, you know, we actually don't measure the abundance of anything. We measure the relative abundance of things because the sequencer itself has a capacity, right? Nobody's ever bought a MySeq run and got NovaSeq data. It just doesn't happen, right? You get 20 million or you get whatever the machine will give you, okay? And so what you're able to measure is the proportions between things as opposed to the actual values. The actual values are what we think, but we're measuring proportions. And then the third reason is that the tools that are commonly used or commonly suggested to be used are not designed for the data that we've got. So even the data that we've got off of the sequencer, the tools that are designed for this, so EdgeR, DESeq, you know, Voom, Lima, these kind of things, they're designed for data sets that are like this, we're going to call this dark blue. This dark blue, we're looking at the ratio or the relationship between relative abundance and the variance or the dispersion. You know, it's a very, very nice tight relationship. And, you know, so that's what the tools are expecting. So the tools are expecting this. They might be expecting that. That's the same relationship for single cell data. They're definitely not expecting this. That's the relationship for the meta-transcriptome data I'm going to show you. It's really a lot more dispersed, still somewhat linear, maybe-ish. But, you know, certainly it's not this. This is 16S data sets, and certainly not that. There's a flat relationship. That's an in vitro selection experiment. All of these are experiments that you can read out with high throughput sequencing. But to try and say that you're going to find one tool that depends upon this exact, you know, that exact relationship to read out the answers for those for different kinds of data, I don't think is realistic. Okay. So we have lots of problems. You know, sequencing is our probabilistic random samples. Their compositions, everything correlates with everything else. The levels are confounded, the bacterial load. And I see Justin's here, so he has opinions on scaling. And I think that's the next thing that I have to do before I do finally publish this data. And, you know, because their compositions also, our conclusions change often with tools that are inappropriate when we include or exclude different sets of features, okay? And then, you know, and that's before we even get to the fact that they're sparse, high dimensional. And, you know, and of course, the biggest problem is that as a species, we're not particularly good at self-awareness. And so most of us are at best kind of begrudgingly aware of the problems and you know, try to, you know, it works well enough. So we'll just live with it. So let me try and give you an idea of what the problem actually is. So let's just say that we're sequencing, we only have three or four things, tigers, ladybugs, and space aliens. And we generate random counts. And this is kind of what we look at in a multivariate sense, you know, use pairwise, form a cube, shall we say. And this generalizes to as many dimensions as you can imagine and beyond what you can imagine. And, you know, if we generate some random uniform data, that's what it looks like. Nothing's correlated with anything. As soon as we convert it to a composition, if we take that data and we subsample it or we sequence it, this is what it looks like. The two most abundant things are negatively correlated. Enterotypes, anyone? And other things, you know, the correlations change in the other ones. So this is the kind of thing that you should make the hair in the back of your neck stand up if you're trying to understand what's going on in the environment using high throughput sequencing. So you have to change the answers that you're trying to get. And the problem is that the data cube collapses down into a simplex where we have one less dimensions than we have variables. And that's the fundamental problem. And it doesn't matter if you have, I'll show a data set that has 450,000 genes, 450,000 potential dimensions. I still have one less dimension than I have variables. I'm still on a multi-dimensional simplex. Okay. And then, so we'll just normalize our way out of this. Nope. Again, here's the data that we think we have. We convert it to a proportion. This is the DESeq transformation, the relative log expression transformation on that data. This does not look like that or that, right? So geometrically, we are not able to fix this in any realistic way. So I'll just say that any transformation, I've chosen one, any transformation that you want to choose from the basket of transformations does not recapitulate anything, any either the numeric or the relative data in any way, shape, or form. Okay. It's most obvious when you're looking at the most abundant things in any sample, but it persists no matter what features you're looking at. Okay, so a semi-general solution, I would say, is using log ratios. This is something called compositional data analysis. And so the log ratio does not move the data off the symbols. Okay, you're still there. And you can use the geometric mean. So what we're doing is we're taking for every part, every gene, every species, whatever it is, we can divide it by the geometric mean of, in this case, all the parts. This is the standard log ratio, all the parts, one part or a subset of parts. And the idea is we get a ratio. I tend to think like a wet lab person. And so what we're really doing, if you want to have a wet lab analogy, what we're trying to do is we're moving the high throughput sequencing data into a large multivariate qPCR kind of experiment where we're looking at this relative to some internal standard. And it's, the denominator then is what's our internal standard. Okay. And I'll just mention parenthetically that the relative log expression and the TMM that are used in DESeq and EdgeR are themselves ratio methods, although they pretend to return counts. They're also ratio methods. Okay. As a teaching tool, I built something that's on CRAN now. It's called AMI Compositional, or AIC, which allows you to examine your particular data set with with all the transformations that are out there, and ask the question, if I change the filtering, do I change the distances between parts? Is what I'm getting is a transformation what's called dominant. Most transformations turn out to be dominant, which is nice. Are they scale invariant? If I get answers from a MiSeq or a HiSeq, do I get the same answer? And it turns out that the RLE and TMM transformations are not scale invariant. They're terrible at that, actually. If I have batch effects or perturbations of some sort, how do they behave with batch effects? And it turns out that most of those transformations don't work all that well. They do behave well in some data sets. So like transcriptome data sets or single cell data sets, they work beautifully because, again, that's what they're designed for. 16S data sets or Meta-transcriptomics data sets, not so much. So log ratios behave rationally under all scenarios. And now just a brief interlude on correlation. It's common in the field to do network analysis using correlations between your parts as a way, as people think, okay, this is kind of a way around this whole problem. We'll do network analysis. So so this is, bear with me for a second. This is the correlation coefficients for the correlations observed between parts for 4 different kinds of data sets. But what's important here is the size of the data sets. So Leonard Foster published a paper in Nature Methods, I think about a year, 2 years ago, looking at, how do correlation approaches using all the different transformations work in single cell data? How well do they work? And it turns out that they're all terrible, including the compositional correlation approach of calculating proportionality. They're all terrible. So it turns out that correlation in high throughput sequencing data sets at low numbers of samples is driven largely by the transformation. So this data set has 7 samples. And at 7 by 7 samples, the correlation between the correlations observed with different transformations, the minimum correlation is about 0.18, and most of them have correlations around 0.2. So most pairs, so if I look at the correlation between the standard log ratio and the RLE, it's about 0.3 as an example. It's the correlation between the correlations that are observed. If I up my counts to about 25 samples, the minimum correlation observed is 0.33. If I up the number to about 50 samples, the minimum correlation observed between different transformations is 0.6. This is about 150. Minimum correlation is 0.91. If I'm doing correlations at single cell level where I've got 1,000 or 10,000 genes, the correlation between correlations, no matter what the transformation is about 0.98 or 0.99. So when you have, the bottom line is when you have a lot of samples for correlation, you're going to get the same answer no matter what you do. If you have a small number of samples, your weighted correlation network graph is going to be driven entirely by the transformation used and not by biology. That's just a side note. I just found it a couple of weeks ago. Okay, so we've developed a tool that we call ALDEX. This was developed by a previous postdoc of mine who was a cryptographer before he came to the lab, and now he works at Shield AI as a lead data scientist, and they build AI systems for military drones. But he says we're never going to drop a bomb on anybody, so it's okay. He's just abstracted himself completely away from the military application. But so ALDEX is a tool that does not do a lot of things that get you into problems. Okay, so it doesn't estimate parameters at all, right? It doesn't assume that your data are any distribution. It doesn't pool information across features. So in order to do the normalization, EdgeR and DEC do pool information across features. So you get that. So if your relationship between abundance and variance is really tight, that's not a problem. If it's all over the map, that's a problem. It doesn't subset the data, and it does not do correct for sequencing depth. Okay, so, but it does take the data and estimates the probability of observing that data. It gives you a probability distribution where each instance that we collect is consistent with the data that you observed, even though if it's not identical to it. And the variance, that correlation problem is driven entirely by the fact that the variation at the low count margin, and by that I mean anything where the count is less than 100, is monstrous. It's exponential, basically. It does account for measurement error, and we return a mean ensemble value. And you'll often see, or you will see ALDEX compared to other tools in this. And it's like, this doesn't control for the false discovery rate problem. It's way too conservative. Well, the expected value of a p-value is random uniform if there's nothing going on. The expected value of p-value is calculated from multiple instances of the data is not 0.5. The expected value of that is actually not uniform. It's actually 0.5. So we're not actually trying to estimate point estimates. We're trying to estimate ensemble estimates with all of it. So in the background, just to make that a little bit more concrete, if our data is the front one, what we're doing is we're generating from a Dirichlet distribution, which is actually a conjugate distribution of the negative binomial that's used by EdgeR and DEC. We're estimating probability distributions that are consistent with our observed data, but are not identical to it. We then log ratio transform all of those probabilities. We calculate a test value for each of those instances, correct for FDR on each of those instances, and then report the expected value across the whole set of instances. Okay. What we observe is that just basically throws your false positives out because false positives are due to random variation, and anything that is actually driven by the biology still tends to come forward. What we typically see in data sets when we benchmark is that essentially this approach gives us the intersect of EdgeR, DEC, VUM, and Lima, okay, which is something that people often do. So again, under the hood, if we had a point estimate, that point estimate gets converted to a distribution, two different groups, two different distributions. From those two different distributions, we can calculate a pooled dispersion estimate, a pooled difference. These are non-parametric estimates. They're not, they're not differences in means and differences between and standard deviations, but they're they're linearly related to those. And then from that, we can calculate an effect size, t-tests, all that kind of stuff. Okay. So we have a tool that's somewhat generally useful. Doesn't make a lot of assumptions about the data. So now let's look at it. So meta-transcriptomics. So awful data, like not difficult, they're awful data sets, honestly. You're sequencing all the mRNA in a collection of bacteria. You want information about what's being made now, and ideally you want to correlate that with, say a metabolomic output collected at the same time. They're often really unbalanced. So you have situations where different conditions have different taxonomic compositions. As I said, everything changes. And as I said, also relative abundance doesn't necessarily reflect the environment and reality. Okay. So naively, when we started, we thought, okay, we're going to choose a nice simple system. We're going to choose bacterial vaginosis, which is a common condition that is it's a common condition where we have a situation where the healthy condition is primarily lactobacillus species, or whatever they're called now by the taxonomists. It'll change tomorrow, so don't worry about it. Or BV is a mixed bag of anaerobes with lactobacillus iners. We thought this would be really simple because it's like a dozen organisms, dozen or 20 major organisms. But of course, we chose the worst possible scenario because the order, there's about 3 orders of magnitude more bacteria in a BV sample than a healthy sample. 3 or 4 orders of magnitude more on average. So huge asymmetry. And this is a situation where there's a big scale difference, and that's something I know that Justin's been working on. There's also not just an asymmetry in bacterial abundances, but a huge asymmetry in which functions are there. So lactobacilli don't make a lot of their own metabolites, whereas the anaerobes make everything, right? So we group things to functional levels, and we have to make the assumption that something stays the same. So there's been a few vaginal metatranscriptomes published. The example I'm going to use is a small one that was published a number of years ago now, where the big conclusion was that what's driving the difference between the lactobacillus and the anaerobes is the relative abundance of ribosomal protein genes. Ribosomal protein genes don't encode anything that, anything pathogenic. They don't encode any genes that are, I mean, basically what they've done is they have, with their data set using the tools that they use, they basically just said, you know what, we have a lot more of these than those, and we can measure that by measuring the ribosomal protein levels. A lot of money spent, not a lot. So the data sets that I have, so this is our vaginal swab data set. These are treatment naive women, and we have a vaginal wash data set that was published by Deng in 2018. And these are healthy, and all the healthies are post-metronidazole. So I started with a nasty data set where we have a lot of variation, and I'm using as an example, I'm trying to find even the worst possible data set because we've got 2 different collection methods, and treatment naive and post-treatment data sets. So massive batch effects between the 2 groups. So can we still get something from this? Okay. So just for reference, this is what you get if you take this data set and run it with EdgeR. You get the same kind of thing if you run it with DBC. So this is a bland-Altman plot or an MA plot. Each point is an individual function. So I've grouped genes like, glucose 6-phosphate dehydrogenase. I've grouped all of the genes that are that particular function into one point, no matter what species. Okay, so that's a functional grouping. Red are differentially expressed. Black are non-differentially expressed. And there's a couple of weird things about this. Okay, so first off, the middle of the data is about at the y-axis is about log 2-fold change, difference between of about log 2, so 4-fold change. The middle of the data down here is approximately even. So our data set is actually canted. right? That's not a good thing. The second thing is that the assumption that's being made by the TMM in EDGAR, the RLE and DE-seq, and by the CLR is that most things are invariant. So if we draw a line of fit through the maximum density of the data set, that doesn't hold. Most things are, actually most genes are variable, okay? Again, that's a bad thing. So this is not fitting any of the assumptions of any of the tools out there. I showed you that the dispersion and abundance plots look great. And then the other thing that they're doing is that a zero is always zero. And so that's why you get these lines here and why you get these straight lines here. Parenthetically, over the years, so I've been following the approaches of different tools and benchmark them against data sets over the years. Tools that are in Bioconductor regularly change how they treat zero values. Okay, and so if you take a data set that's relatively sparse and analyze it today, and you analyze it in 3 years, or take a data set that was analyzed 3 years ago and analyze it today using the exact same tool that has not had a major update, you will get different answers. Because they do change how they manage zeros, sometimes for computational efficiency, sometimes for, I don't know why. Okay. So one of the biggest things we need to do is we need to get rid of that canted data and we need to center it appropriately. We need to figure out what it is that's invariant. We need to choose something that is important, right? Or that we think is a good reference to use. So the CLR uses everything is in the denominator. That's not a good reference because not everything is, at least half of it's not invariant. We could choose one special function. I don't know which one I choose, but you would get a slightly different answer, or you get a slightly to completely different answer if you use a different reference. This is called the additive log ratio in compositional speak. In qPCR, that's just qPCR. That's all it is, right? You've got a reference, you've got something that you're measuring relative to that reference. We have a couple that are somewhat that allow us to choose a reference somewhat automatically. And for this data set, the one that works the best is the low variance, high abundance. So what we're doing is we're choosing those genes that have low variance within each group. We look at the intersect of low variance genes between groups, and then we say they have to be high abundance. So if you think about what that is, transcription, translation, intermediary metabolism, your typical housekeeping genes. So it's a sort of semi-automated way to find housekeeping genes. Okay. So does that work? Yes. So here's an MA plot. MA plot on, again, individual points. And I've colored the low variance, high abundance genes in blue. And you can see that they nicely line up right on the center line. They should, because they, what we're doing is calculating the geometric mean of those values, those count values. They're also nice because they're high abundance, they're never zero. Awesome. They line up dead set on the center as they should, because the geometric mean of those should be exactly zero. And we look at other housekeeping genes that are not included in that basket. And ribosomal RNA and tRNA genes also line up. There are ribosomal RNA and tRNA genes that are barely expressed, and they also tend to line up in the middle. So housekeeping genes, we can find them, we can use them as a reference to center our data appropriately. What also you can see is that at the low count margin, this should read abundance, sorry. At the low count margin and at the high count margin, we're completely straight on. So we've got rid of that cant as well. Okay. So this is then, remember I've got the data. So now this is looking to combine the data sets where I've collected data from vaginal swabs and vaginal washes, treatment naive versus treatment versus metronidazole treated. And so it's a little bit of a busy plot. So up at the top are our swab samples. They're marked with an S. And down at the bottom, our fluid samples are marked with an F. So we can see that we separate those two groups, those two collection cohorts on component 2 of a principal component plot. These are the samples, these are the functions. On component 1, we're separating the biology. On component 2, we're separating the cohorts. And again, you can see those are the genes that we're using as our center to make to center the data. So again, genes that don't change end up, not directing change between the groups or between the cohorts. Genes that do change, so the red ones, I'll show you, the red ones are the ones that are different between health and BV, and the blue ones are different between cohorts. And we only have a few genes that are different in both. So we've been able to separate. And the way that I did that is just put it into a generalized linear model approach inside of ALDEX and calculate the expected values, where I built the model such that I can ask what's different between health and disease, health and BV, independent of what's different between the cohorts. So this is an effect plot. I like to say this is the kind of plot that you wish you had when you had a volcano plot. So what we're measuring here is the difference between groups, and this is the within group variation of the dispersion. This line is the line of equal dispersion and difference, and each point here is an individual function. And you can see that if we're plotting the difference between cohorts between health and BV, so these are genes that are or functions that are up in health. These are functions that are up in BV. You can see that even though our data is asymmetric, by centering on our low variance, high abundance genes, we've nicely centered this, and we can pick out, this is largely TCA and amino acid metabolism, but there's a few other things. And we can exclude almost all the genes that are different between cohorts. So I think that's pretty cool. And you're going to struggle to find a data set that's this wildly odd. So what we can then do is we can say, okay, now is this internally consistent? So we can then, instead of using differential abundance, we can say, okay, let's do some exploratory analysis and see if what we find exploratorily lines up with this. So we can say aggregate all of the genes that we've mapped reads to, and sum across those counts and map those sum across those counts that are mapped back to an individual species. So basically, this is just taking $40,000 of RNA-seq data and turning it into a $1,000 16S data experiment. But we like to do these things. And what we see is that yes, BV separates from health. It looks like we might have a couple of different kinds of health, a couple of different kinds of BV. And what it's driven by is, in fact, the presence or absence of lactobacilli and these mixed bag of anaerobes. Awesome. So this is not a functional level analysis. This is a gene level analysis. So every point here is a gene, is a gene that has at least one read mapped to it. This is mapped using the Virgo database from the Maryland group. They're colored by the gene mappings are colored by the species, the presumptive species of origin of that gene. There's approximately 450,000 map points on this plot. Okay, and this is why I have to run it on my own laptop, because if I transfer this to anybody else's laptop, I guarantee it will either crash it or it just won't display. Okay, and I really need to learn how to make these files smaller, but not a priority. We can see that basically all the healthy samples, regardless of cohort, are associated with lactobacillus genes and not with anaerobe genes, which is basically a nice clear separation between them. And again, it looks like at the gene level, we have maybe 2 or 3 different kinds of BV. So we can explore down a little bit more. And this is only the those same genes only with the BV samples themselves from the two different cohorts. And it looks like there's, on component 1, it looks like there's basically two kinds of BV. This type on the right, this type on the left. The right ones tend to have, looks like they have a lot more prevotella. So this is amni, this is buccalis, and one is square bivia. Yeah, so buccalis and bivia, amni, and then the ones on the left, it looks like there's a lot more Gardnerella, relatively speaking, although there's 1, 2, 3 clusters of Gardnerella. Gardnerella fruit, I believe, is broken into multiple different species at this point. So, some evidence that there's multiple different kinds of Gardnerella in this sample as well. So is there any functional difference between these two types of BV? So this one tends to have actually a more mixed bag of species, different prevotellas, different, and there's megasphere in here, and there's something that's unknown, unmappable. BVAB1, I think is what it was called, or no, it's this one. I can't even pronounce it. And it's the names, I'm sure the name has changed anyway, so it doesn't matter. And then a cluster over here where you've got a lot of Gardnerella, but almost not as many of these anaerobe genes. We can then ask the question, what separates those two groups? And it turns out that the group on the right that has more of the mixed anaerobes has a lot more genes for motility and chemotaxis. So this is each point here is a function, and we're measuring the median effect size for the difference between groups using ALDEX. So this is the effect size, the difference between divided by the effects, by the dispersion. And then they're colored. If they're red, they're up in BV1, blue, they're up in BV2, the ones right and left. I've switched the axes, I'm sorry. So the motility and chemotaxis is much more prevalent in the one on the right that has a mixed bag of anaerobes, as is arginine, uricycle, and polyamine, and amino acids and derivatives. And so this you might want to make the case that the BV on the right that had multiple types of anaerobes, this type, is a more problematic BV because that's the one that has a lot more species that have motility. This correlates also with more biofilm, more of the fishy odor of BV, whereas this tends to not correlate as well with those. So we've split BV by transcriptome. And looking at the healthy ones, again, really nothing new that we didn't know from 16S. Crispatus driven, inners driven, and we have one sample that is maps exclusively to Jensenia, so just a different line of facilities. So bottom line, going back to our original mixed, our original examination, we're seeing multiple different kinds of health, multiple different kinds of BV. Internally, what we're seeing is the differential abundance lines up almost perfectly with what we soon do exploratory data analysis, which is nice. Yeah, and so the one thing that I still have to wrap my head around is the fact that when I'm looking at this cohort, is that when I'm looking at relative abundances, yes, we can separate health and BV really well, but this set of genes corresponds to 1% the abundance of at the species level relative to the abundances of the species that are in that cohort. So it's given the fact that there's a 2 or 3 log difference in actual abundance in the environment, I would have hoped to have seen a much bigger split still. We're still not analyzing this to its full potential. But we're getting there. Okay. So what have I tried to tell you? Well, metatranscriptomes are hard. Okay. But at least I think we're approaching a point where we can get reproducible answers across, I should have said cohorts instead of data sets. Okay. I think that's a win. This data set that I've shown you is way more variable than any data set that you'll get from, like from gut, for example, or maybe not skin, but certainly from gut. Okay. It's actually asymmetry in data sets is surprisingly common. So I chose a data set because I really don't care where the data come from. I chose a data set that's really hard. But asymmetry is also surprisingly common no matter where you're looking. Okay, so if you're looking at, we have a project on wastewater treatment at an oil refinery, and you would think, it's the same oil coming down the pipeline. If we collect sequentially, sometimes we get data sets where we have not a single organism in collection in June of 2020 is the same as in May of 2022. Like not a single organism overlaps. Okay, and that is ostensibly it's the same. It's the same. It's the same feedstock. It's nothing's changed, but asymmetry is amazingly common. Of course, in the gut we have enterotypes. We have that continuum of one thing or another is the dominant species. Proper centering is crucial to prevent nonsensical answers. Okay, if you don't center your data, it's kind of like, I don't know what it's like here, but in Canada, every kid wins a gold medal. Like every kid that does a cross-country run, first or last, 3 minutes a kilometer, 30 minutes a kilometer, everybody wins, right? Until you get to travel teams, and then of course it becomes a little more competitive. But unless you properly identify those things that are truly different and those things that are truly the same, you're just going to get nonsensical answers. Unless you center it properly, you're not going to get right answers. Or let me rephrase that. I'm not saying that what I've shown you is correct. I'm just saying that it's internally consistent. Okay. ALDEX, it makes this probabilistic distribution of the data, and it assumes that something is invariant, and sometimes you have to choose that manually. And it appears to be what I've shown you appears to be a fully general method. So I can take a data set from ENA and reanalyze it, and teach it to a class, and it's like, oh yeah, we can recapitulate all of those steps in a fully generalized way. So Andrew is the guy to blame or thank for ALDEX, and for getting us started down the compositional analysis, the log ratio approach. Gene collected a lot of the data and had done a lot of preliminary work. My microbiome plays a big role in who I am, of course. Justin, so the tiger ladybug, the space alien analogy, I stole shamelessly from Justin. I saw him make a presentation on that, and I thought, damn, that's a great idea. So I've used it shamelessly ever since. And I've had several other collaborators and some funding along the way. And yeah, that's basically it. Anybody whose head hasn't exploded by now, we can have questions and I can explode your head if you want.
Thank you so much, Greg. This was awesome. We like to have the first question come from a trainee. Anyone?
I have a simple comment about it. Great talk, thanks. This is after the mapping and generating the counts, right?
Yes.
Are there any problems associated with meta-transcriptomics before, while doing the mappings and getting those counts? For example, Virgo uses their own databases, I think.
Yeah,
right?
they do.
Yeah. So do you think it's also dependent on the type of databases you're using?
Okay, so great question. So what we have done, we've had this data set for a long time. So what we've done is initially we took this data set and we mapped it against a data set that's called SEED, the SEED data set database. Gene probably spent 2 months downloading the SEED database because it's not actually publicly available. We have it though, if you want it. So we did all of this with seed, and we mapped about 30% of the reads to the seed database. We got exactly the same coordination. We got exactly the same difference between health and BV, the same. So we did it with Virgo with the 2 data sets, and we're mapping about 60%, 60 or 70% of the reads. We get the same ordination, we get the same separation. We also independently assembled our RNA-seq data, annotated that, and we got the same damn answer. Okay. Now that's not to say that that it's identical. I mean, it's we're getting we're changing the percent variance explained. We're changing the separation and the exact placement of things. But the big picture is that we do get 2 types of BV driven by chemotaxis and motility and polyamine biosynthesis and degradation. We do separate the health into multiple different types, So like the big picture is the same, and it's not driven so much by the front end as by the back end.
Thank you.
Any other questions? Reminder to the trainees, there will be lunch after this. There.
So it's really impressive how much you've been able to do with data sets. But my question is, if you had done the same thing with just metagenomics, would you have gotten the exact same answer? Is there actually gene expression difference that's driving this, or is it just the present absence of these different functions?
Yeah. I don't know. I honestly don't know the answer to that. Because we don't have a metagenomic data set to compare to. Okay. We do get a surprisingly similar answer if we do collect 16S and then just do imputation. However, what we don't get if we do that is we're not able to separate the BV groups. Okay. So I think, if I was to guess, I would say that the difference between health and BV is driven entirely by the difference in abundance of the species. I think that's what that is. Okay. Internal to BV, I think that that there is value to the meta-transcriptomic because we do have that same population of organisms, and it's not driven by presence absence. It's driven by changes in the abundance of the organisms, and then we also then do have different transcript levels in the same organism in different things. Okay, which is so that's contributing something to it. Whether it's the major driver, I don't know. Okay, so yeah, I mean it's a great question, and the answer is we don't really know. What we do know is one of the things that we do get from this that we don't get from metagenomes is that we do see, I didn't show it, but we do see interspecies cooperativity. So we see that we have pathways that are incomplete in different species, where if you connect up the species, you then can connect through the pathway. And we have one example where we have, and again, I didn't show this data because that was published by Amy. We have one example where a metabolite is produced that can only be produced by passing the intermediates from one to the other, and that correlates with the expression in those species in the samples that we actually see that metabolite. And so maybe you've been more impressed by that, but
no, yeah, that's really cool, actually. Yeah, that's something that I'm really interested in. But for the motility then, so if you see the difference in motility like within a species, do you have any hypotheses about what would be driving that? Why is it more motile under one set of circumstances?
So we think that the difference in motility, going back to your first question, actually, we think that the difference in motility is driven by the presence or absence of one or two species. So most of the species in there are non-motile. In the one on the right-hand side, there is the presence of species that are motile. The polyamines, it's actually the polyamines that we have that's metabolomic. Polyamines are driven by gene expression differences in different species, but the motility is driven by presence absence of the species.
I have a question from Zoom. If you did have a metagenomic data set to compare to and wanted to detect actual changes in gene expression, would you use essentially the same approach to analyze both data sets? Any special considerations?
Yeah, I'd use exactly, I mean, I would use exactly the same approach. It's, as I say, that it's a fully generalizable approach. We use the same approach for 16S. I showed the example of those 5 different data types intentionally because I take this exact same approach. It's single cell, it's 16S, it's cell X, it's transcriptome, it's metatranscriptome, it's metagenomics. The same approach works for everything. Any kind of sequencing output. It also works beautifully for proteomics data too.
With
Yes.
metabolomics data?
Right.
With metabolomics data?
Yeah, if you're running a metabolomics data set where you can saturate the machine, then you've got the same kind of problem. And so you can use, we have used the same approach of using an internal an internal standard as a denominator for metabolomics data. And it also made a big difference.
Justin.
So I really like that correlation slide where you were comparing the different normalizations. My question is, it seems, so I think you said that as you got more data, the effect of the normalization didn't really matter, and that all of the results tended towards that everything was completely correlated, or
Everything, so yeah, so as you increase the sample size per group that you're calculating the correlations on, the correlations that you observe are less driven by the transformation and more driven by the data. What I'm not saying is that the correlation that you observe at the end is the correlation that you would get in the pre-sequencing data in the environment, because the paper that Leonard Foster put out showed that basically in single cell data, all correlation metrics are able to correlate with known metadata roughly equivalently. So post sequencing, the correlations are you can get them to be concordant no matter what transformation you use, but they're not particularly useful.
so they're not necessarily right, but
They're not right. They're definitely not. The majority of it, yes, they're definitely not right. Yeah, unfortunately.
Thanks.
Yeah, so like even the proper or any of the other compositional association measures don't behave any better than Pearson correlation coefficients or Spearman's or any other correlation metric when you're trying to correlate the the gene expression level to the known biology of the system.
This is something we've been seeing in lots of other systems, in lots of basically every analysis you can imagine of sequence count data.
Yeah.
The more data you get, the more everything agrees, the less right it is.
Yeah. Whenever I see a paper that does high throughput sequencing with correlation, I just don't bother reading it anymore. It's just not worth it. Sorry. Did I say that out loud?
So I will have you here.
So do you recommend before acquiring the data, something specific to help? Like I'm thinking about maybe spiking it, like the standard, so that then for a fact that should not vary, and then that can be used as standard. Okay.
Yeah. So I mean, how much money do you have?
Well,
Yeah. So yes. So people advocate for spike-ins as a way to help get rid of this, right? So there was just a paper spiking in metagenomics a few months ago. And spike-ins have been used in the RNA-seq world forever.
Yeah.
So what I should have shown, but I could call it up, but so the transcriptome data set that I showed that had that beautiful difference dispersion plot. So that's a beautiful experiment that was done by Jeff Barton's group at UBI. It's like they spent a stupid amount of money to do a yeast transcriptome experiment because they were hoping to use it for something else. The experiment is beautiful. It was wonderful to watch what happened to that data as it progressed through the bioarchive. So the experiment was originally done to prove that spike-ins were going to solve this problem. Okay. And then it became, the first axis of variance is the difference between the wild type and the knockout gene. Second axis of variance was the batch of the spike-ins that they chose. So, I mean, if you're going to do spike-ins, and I have seen spike-ins that have been done very well. If you're going to do spike-ins, you actually have to design it in such a way that you have zero batch effects with your spike-ins. So you have to have enough spike-in to use across. If you're going to design an experiment that's going to be a longitudinal experiment over 10 years, you better have all your spike-ins ready ahead of time, know that they're stable and so on. You're going to have to put them in at relatively high levels. So you're going to have to sequence more deeply, spend more money. And you're going to have to make sure that you have enough spike-ins that you can independently estimate, the actual value that you want to get, because what you get from sequencing is a point estimate. And those point estimates can vary quite wildly. So you need to spend a lot of time and thought to get the spike-in protocol correct. And by and large, most people just don't have the patience for that. So yes, I can't do it. There's no way in the Canadian system that I could ever get the money to do that. In the NIH system, I think you guys have a little bit more wealthy. The consortium approach, I think it's probably a good idea. But again, if you change the batch number, then it's all been for naught.
But what is the role then of targeted confirmation? Something like a qPCR assay to help benchmark some of this data.
Yeah, so I mean, a qPCR, if you're using a log ratio approach, you're not doing anything different than you would be doing using qPCR.
I would assume it wouldn't have all the plethora of problems though of a large sequencing.
Well, all you're doing is you're basically, I mean, you can use qPCR to validate what you've seen is actually real.
To get to accuracy.
You'll get a more accurate value from a qPCR, but it's not truly independent unless you're doing it in an independent cohort. If you're doing qPCR in the same samples, you're just adding a zero to the to the decimal
But if
place.
you want to know your sequencing run is quasi accurate, you confirm it with that method and then use a separate cohort to validate.
You could use, so I mean, all of this is exploratory. I mean, it doesn't, to me, I don't think it explains anything, but it gives you the hypothesis that then you can go and look, okay, I've got this set of 5 things I'm interested in.
Yeah.
Is it a reasonable biomarker so that you can metabolize, for the genes that are in the pathway, or you can do qPCR or some kind of, as a way of it.
Yeah, totally agree.
Within the same cohort, you should get, within the measurement error, the same answer from qPCR as you got from high throughput sequencing if you use the log ratio approach.
All right, we are 1 minute past. There is one short question from Zoom. Is there any scenario where CLR would likely work better than LVHA?
Yes. So there are data sets where the LVHA doesn't work. So in vitro selection is even more asymmetric than Meta-transcriptomics, and it doesn't work. Okay. So we have other ways of finding midpoints that are useful, but they're even worse to analyze than this.
Right. Let's thank Greg.
That's